I'm Gail Robertson. I'm the
hi.
president of the Biophysical Society.
Oh, wonderful.
And I
How are
think
you?
we haven't had the opportunity to meet yet, so. Or maybe we sat near each other.
We sat
at
near
that-
each other at
that
the
table.
last Biophysical Society. Yes, that's right. Yes, no, I recognize you. That's why, yes.
Yes.
And yeah, well,
Very happy to have you here.
Thank you very much,
My
because
whole lab is coming. They're running late.
it's a wonderful place. It's,
Yeah.
I mean, the biochemistry and the biophysics in this place is amazing.
Yeah.
I was just visiting the cryo-electron microscopy facility. It blew me out of the water. I
Wow,
mean,
wonderful. Yeah.
we wish we could have something like that at Berkeley.
Well,
Anyway,
good to see you.
thank you for
Yeah,
coming.
Thank you
All
for
right.
coming
That's
to
it.
Madison.
Thank you.
Hello.
Should we wait a couple of minutes? Because I see some people are still. Huh?
Do you fly up today?
No, tomorrow.
Tomorrow.
Yes, and I go to Washington D.C. because I have to go and pick up my grandson, who is with his grandmother, my wife, visiting our son, and her little cousin in Washington, and my wife took him there 5 days ago, but now he has to come back to California with his parents, except that there was nobody that could actually bring them back, and so they told me, "You are in Wisconsin, go to."
You're already halfway there.
So that's what I'm gonna do tomorrow. Go to Washington first and pick him up there, and then fly with him back to.
Are you staying?
No,
Just going.
just 3 hours in Washington, and then take a plane.
How long is the flight from here to Washington?
That's a good question. I think it's probably 3 hours, maybe 2.5 hours, 3 hours, but then the other one is 6 hours. Yeah, from Washington it's more like 6 hours. Yeah, so we'll see. Yeah, well, I'm crazy for that boy, so me tiene loco. Me dice Tata. ¿Dónde está el Tata? Ay, Tata.
Mi abuelo, mi papá Humberto, le llamo papá Humberto y él le llamaba Pata.
Pata.
Mi Pata, vamos a comprarlo.
Yeah, and CJ sent me a Slack to remind you to introduce yourself.
Oh, okay. Great. Thank you. This is the other undergraduate that I'm working with, Victoria.
I see. Were you in at lunchtime? Yeah. I saw you there. Yeah.
Next year she'll be applying to grad school.
Well, it's cool.
Yes.
Very good.
Terrifying.
Yeah. That's okay. Many people do it all the time. Okay. You see? Okay. Yeah.
Hello?
So I think we'll go ahead and get started if everyone's settled in. The mic's not very loud. But my name is Bianca Chavez. I'm a graduate student in Dr. C.J. Lim's lab. And today I have the pleasure of introducing Dr. Carlos Bustamante. to the University of Wisconsin-Madison's second lecture for the Harry Steenbock lecture series. So first I want to thank the Harry Steenbock Endowment sponsors who made these talks possible. And as always, we recognize Dr. Harry Steenbock, who's a distinguished professor at the University of Wisconsin-Madison, for his outstanding achievements in nutrition and biochemistry. So Dr. Bustamante is an HHMI investigator at the University of California, Berkeley. He received his master's in biochemistry from the National University Mayor de San Marcos in Lima, Peru. Then he went on to the University of California, Berkeley to earn his PhD in biophysics under Dr. Ignacio Tinoco. Dr. Bustamante is a pioneer in single molecule biophysics and who specifically focuses on single molecule manipulation and detection methods. He was a recipient of the 2012 Vilek Prize for Biomedical Science, which recognizes foreign-born scholars with a lifetime track record of achievement and innovation. He was named one of America's Best by Time Magazine in 2001, and he was also elected a member of the National Academy of Science and American Academy of Arts and Science. So today we have the great honor of hearing Dr. Bustamante talk. Tell another story about his lab. So let's welcome Dr. Bustamante.
Thank you. Thank you. Thank you very much, Bianca. Bianca and I have the connection that actually her father is Peruvian, so we Peruvians are everywhere. Peruvians are the first export of Peru, right? The first product of export of Peru. Anyway, thank you again. Well, thank you for being here today. I'm going to tell you a different story today, in which, again, we are going to use these methods of single molecule force spectroscopy to try to answer a few questions about how is that proteins fold as they are being actually translated, as they are being synthesized. And I said two stories, and I'm going to start with the first one, which involves the analysis of how a protein folds in real time on the surface of the ribosome. So, as you know, proteins begin to fold as they are actually coming out of the ribosome, but most of what we have learned about protein folding has been obtained from what we can call in vitro refolding of molecules that are completely synthesized. And so the question is, how does this in vitro refolding differ from de novo folding, which is through proteins actually being synthesized on the surface of the ribosome? We know that some of the protein stability is affected by the presence of the ribosome surface, and that the rate of refolding sometimes is greatly reduced on the surface of the ribosome. In fact, for example, here you have one study that shows that the concentration of the mid-urea concentration for unfolding a protein like DHFR is greatly reduced on the surface of the ribosome compared to when the protein is just in solution. And in our own laboratory, we actually show that the refolding of a protein like T4 lysozyme actually near the surface of a ribosome is reduced by more than 200 times relative to the protein just folding in solution. So what this is telling us is that the surface of the ribosome is not an inert surface, but that actually somehow is able to interact with proteins and affect the kinetics and even the stability, thermodynamic stability of proteins. Now, we may ask, however, are there other differences? And one immediate difference that we can think about is that when you are off the ribosome with a fully synthesized protein, the potential energy surface over which the protein must diffuse in order to reach the native state already pre-exists to that native state, right? It already exists because it's encoded in the sequence of the protein. Whereas for proteins that are being synthesized on the ribosome, the potential energy surface is being generated as the amino acids are being added and the information is being encoded in the sequence. And so the question that we asked was, Does the folding pathway of the protein change completely in this case versus that case, right? Displaying perhaps different intermediates or occurring at different times during the folding process, right? So that's what we wanted to ask and answer. And so one interesting observation when thinking in terms of protein folding, particularly co-translational protein folding, is the realization that the protein synthesis is very slow, typically at the most 10 to 20 amino acids per second, and even less for eukaryotes. And whereas folding is relatively fast process, it can occur in microseconds or in milliseconds, right? And so because of that, it has been assumed that because the protein folding is so slow, it has been assumed that the folding of the growing nascent chain has enough time to equilibrate with its ensemble of accessible conformation after every amino acid is added, because the amino acids are being added so slowly, right? Okay, so one inference from this assumption I wrote here in green, one inference from that is that it should make no difference whether we study co-translational folding in real time or whether we study them using stalled, equilibrated ribosome-bound nascent chain complexes. I'm going to use this abbreviation for ribosome-bound nascent chain complexes, RNC complexes. When you have a ribosome-bound nascent chain, of course, you just generate them, the ribosome is stalled, and then you just wait until you eventually can grab the protein and you can study the protein folding and folding with your optical twister, for example. But these systems are equilibrated because a lot of time has passed since you first formed them. So the questions that we want to ask are, how does the ribosome affect the folding of proteins, particularly multi-domain proteins? Does it change the folding path? How does the ribosome affect misfolded states? Is the folding pathway different with equilibrated stall complex versus active translating complexes? In other words, is co-translational folding indeed an equilibrium process as it has been assumed? In order to answer these questions, we decided to study the folding and folding on the surface of the ribosome and by itself in solution of this protein called calrethrin. Calrethrin is a calcium-binding protein, 177 amino acids. It's a bacterial protein equivalent to calmodulin. Has 4 EF hand motifs. Each EF hand motif binds a calcium ion, except the second EF hand motif that cannot bind calcium. The first 2 EF hand motifs constitute the N-terminus domain, which is 90 amino acids long, and the third and fourth EF hand motifs form the C-terminus domain, that is 87 amino acids. And so we decided we wanted to study this protein, and in order to study the protein, we wanted to go from unfolded to folded and from folded to unfolded, and we decided to do an experiment such as this, where we actually attach to the N and C terminus of the protein pieces of DNA, fragments of DNA, which we call DNA handles, and then we attach those DNA handles via, for example, biotin streptavidin to beads, each one of which is actually kept in an optical trap, in an optical twisters instrument. And so it is possible to induce the unfolding of the protein mechanically simply by moving one of the traps relative to the other, exerting tension on the protein, and the protein eventually will actually unfold. Now, I want to say that because the DNA itself has its own elasticity, we have previously, many years ago, characterized the elastic response of DNA, which is very dull, very, very monotonic, and essentially the curves, if you plot the force here versus extension, look like this way, highly nonlinear, by the way. If DNA were a very Hookean spring, namely a linear spring, then force versus extension will be a straight line. but it's not. It really has this shape, this type of shape, okay? So you're going to see, in fact, the behavior of the DNA when we start pulling in the next slide, but you will also see that at some point there is a sudden discontinuity which corresponds to the unfolding of the protein at certain force. So that's what I want to show you here. There you are. Here is the DNA behavior, then the unfolding of the protein, and then we can return, and then you see that the protein actually is refolding. It unfolds in one step. You can see that it unfolds in one step, but then it refolds in two steps, right? So it refolds via an intermediate, and so we can, in fact, study the unfolding and the refolding of the protein, as you see here. Now, if you look at here, you can see that the protein is starting to refold and then unfolds again, refold, unfolds, refold, unfolds. So the system is bistable in this region, and so it would be interesting to study that system here in this region where the protein is bistable. In order to do that kind of study, we can do a different type of experiment, which is, we call it a passive mode experiment. In passive mode, we apply certain initial tension to the protein, as shown here, and then we keep the position of the traps, of the laser traps, fixed. Then the protein is originally unfolded, as you see here, but then the protein decides to cross the barrier of refolding, and the protein refolds. When it refolds, it will pull the beads out of the center of the traps a little bit, and that increases the tension, the force that the protein is experiencing. And after a few seconds, the protein unfolds again. And then after a few seconds, the protein refolds. And then what you will see is this bistability, this transition between the folded and the unfolded state. And so when you look at, this is what I just said, right? When it's unfolded, the protein experiences low force. When it is folded, it experiences high force. And the protein goes between these two states. And this is what we observe. The force versus time this time, you see that the force is being measured by the optical tweezers machine, and you can see that it actually goes between low force, unfolded, high force, intermediate, low force, unfolded, high force, intermediate, etc. And eventually it goes into the folded state, which is a match. So this is intermediate. You see that there are three states: unfolded, intermediate, and the folded state, right? You can actually see the three. And you can actually use a hidden Markov model to actually determine the lifetime of the unfolded state, the lifetime of the intermediate, and the lifetime of the folded state, right? Now, why do we care about these lifetimes? For example, here, the lifetime of the unfolded state going to the intermediate that we saw in the original curve I showed you, right, when we were pulling, the lifetime, you can get, if you get the average lifetime of the unfolded state, the inverse of the average lifetime gives you the rate of refolding. So we can actually obtain the kinetics of the process, right? And the same thing here with the lifetime of the intermediate, and so on and so forth. Not only that, notice that in this type of experiments, you can tell immediately that the intermediate is an on-pathway intermediate to the folded state, because the system attains the folded state passing through the intermediate. So the intermediate is on pathway, not off pathway. But if it would be off pathway, you would be able to notice that as well. Okay, so we showed that actually this dependence on force actually can be written in this way, where the inverse of the average lifetime is proportional to the rate of unfolding, right? And again here, the average inverse of the average of this lifetime of the unfolded state is equal to the rate of refolding, okay? And that is proportional to a term that looks like Arrhenius, but instead of having the activation energy here, we have the force at which this rate is being determined times the change in the distance to the transition state between that unfolded state to the folded state, or the folded state to the unfolded state, as shown here, right? Okay, so there is an exponential dependence on these rates on the force. So if I take the logarithm of k, I will expect a linear dependence on the force, right? For both rates, for the folding and the unfolding rate. One will increase with the force because it has a positive sign exponential. The other will decrease, the refolding will decrease with the force, right? Okay, so we decided we wanted to know what is the intermediate that we are seeing with cal-erythrin. And in order to answer that question, we actually decided to compare the properties of that intermediate, right, with the full protein, with just studying the N-terminus alone of cal-erythrin and studying the C-terminus alone of cal-erythrin. And we can do the analysis that I just told you about the rate constants for unfolding and refolding of the intermediate, the N-terminus, and the C-terminus. And when you do that, using these expressions, we get this data. As I told you, the logarithm of the rate constant for unfolding and refolding are linearly dependent on the force. And you can immediately tell that whereas the N-terminus is very different than the C-terminus, it is also very different from the intermediate, and that the intermediate coincides with the C-terminus identically. So that immediately allows us to say that the intermediate that we are observing is the C-terminus folding first, and then the N-terminus folds afterwards, okay? So you remember I showed you the curve where you see the intermediate, then it goes in, and then finally the protein folds, right? That intermediate is the folding of the C-terminus. Okay, now, and I think that I wrote that here, therefore the intermediate observing solution of the ribosome corresponds to the folding of the C-terminus. So now comes the very interesting question, and is the following. In the ribosome, the N-terminus comes first, and then comes the C-terminus. But in solution, the C-terminus wants to fold first. So the question immediately is, does the ribosome change the order of folding of the domains by forcing the protein to fold through the intermediate, right? So that's what I actually asking here. In order to answer that question, we actually generated stall ribosome bound nascent chain complexes 135, 146, and 157. And what I'm showing you here are refolding curves, okay? Now, it turns out that, well, the number here that I'm using here is the number of the amino acid at the P site on the ribosome, okay? And you notice that here, where the whole N-terminus is out, well, half of the N-terminus is out. Here, the full N-terminus, EF hand 1 and EF hand 2 are completely out the ribosome. Notice that we see no refolding at all. Even when we let some more of the E3 EF hand come out, we see no refolding, right? And that is very different than what we see in solution. This is on the ribosome. This is in solution of the ribosome. We see the first transition, and then we see the other transition. So here is the end domain, right? The first 90, you can actually see that there is a transition, and that's what happens in solution. But on the surface of the ribosome, we see no refolding. So what this is telling us is that somehow the ribosome is not forcing the N-terminus to fold first before the C-terminus, but not only that, it's not even letting it fold, because of the ribosome it will tend to fold, but here not, we don't see that. So we decided to extend now these complexes, these stall complexes, to 166, 167, like this, where part of the E3 is still in the tunnel of the ribosome, as shown here, 167 and 177. And now we see that the protein is starting to make certain transitions to this state, which actually, as I will show you in a minute, corresponds to 130 amino acids folding. And that is a misfolded state, as you will see in a minute. In fact, if we add a linker of 45 amino acids to make sure that the whole protein comes out from the surface of the ribosome, now you see the folding of the C terminus and then the folding of the N terminus. So in the presence of the ribosome, when you have synthesized, when you are in a situation like this or like that, apparently these domains here actually misfold and form some kind of misfolded state. And I can show you that. Here it is. This is the RNC 177. When we relax it, it forms this intermediate that is actually 130 amino acids long, which is much larger than either the C terminus or the N terminus. And so it is actually a misfolded state. And in fact, when you compare that to 177 with 45, plus 45 linkers, so that the whole protein is out, now you can see that indeed the protein is going through the C terminus, and then it's going through the N terminus, and it actually folds, right? It's folded. So we see that there is actually, and so we decided to see what is this misfolded state, and we suspected that it was a complex made out of EF hands 1, 2, and 3. And so we wanted to compare the EF hand 1, 2, and 3 in solution with the RNC 177 on the ribosome, and you see that they visit exactly the same state. So this allows us to tell that what we are seeing here in the previous slide, here, this misfolded state corresponds to the folding of EF1, 2, and 3, and it's a misfolded state involving 130 amino acids. So then we ask the question, is there a kinetic effect? So let me just conclude, first of all, that what we see is that even though the ribosome, in the ribosome, the N terminus actually appears before the C terminus, the ribosome doesn't force the N terminus to fold first, but on the contrary, it prevents the N terminus from folding, right, by interacting with the N terminus, and it presumably to allow the C terminus to come out and eventually fold first, right? But then, if you generate a stall complex of 166 or 177, you find that actually those stall complexes actually misfold. They generate this misfolded state. And the next question we wanted to know is, in solution, the misfolded state is attained immediately. What about the kinetics of misfolding on the surface of the ribosome in these stall elongation complexes? And so you see that actually what we find is that the unfolding of the misfolded state actually is sped up by the ribosome, right? And the refolding of the misfolded state of 130 amino acids is a slowdown, as you can see here, right? Relative to this is changed. However, the time the misfolded state remains unfolded is still quite short, of the order of 1 millisecond. And the time it remains misfolded is of the order of 1.5 seconds. Therefore, we conclude that the ribosome does not seem to help too much to prevent the misfolded state, at least as observed in these stall RNCs. It helps a little bit, but not enough. So we decided that since we still have faith that the ribosome is not there just to be there, right? We decided that maybe we should actually now study these complexes in real time. And that means that we are going to actually have a ribosome that is translating actively a polypeptide, and we are going to grab the polypeptide as it comes out of the ribosome, and we are going to see what happens with that polypeptide when it gets to 166, 67, which is when we first actually observed the misfolded state. Okay, in order to do that experiment, we have to do this, right? We have to grab, again, with DNA handles, the ribosome on the other one hand, which is interacting with its own messenger RNA. We have to add the full complement of tRNAs and elongation factors, etc. And you are going to then grab the polypeptide that is coming out, the nascent chain, and then with another DNA handle, and then the question is, the idea is to actually have a construct like this, where we added an additional linker here on the N-terminus, and we put 3 valines in a row, so that, and we don't provide the tRNA with valines, so that actually the ribosome stalls here due to starvation, tRNA starvation. I should say that actually initially we put 1 valine, and we couldn't get the ribosome to really stall robustly, so 3 valines actually did the trick, okay? So now we have the stall ribosome, and then we can grab the peptide that is starting to come out and then follow the process in real time. What we are going to do is we are going to do this experiment in passive mode. So what is going to happen is that in passive mode, you start with an initial force, but as the molecule gets longer, as the polypeptide gets longer and longer, obviously that force will begin to sag, right? Because the system is getting longer and the tension will be decreasing. And that's what you're going to see as the ribosome translates. So that's what you're going to see here. You see that the force is coming down, is coming down, is coming down, and eventually it arrives to 166, which is here, 67, and then nothing happens for a while, and then eventually it starts actually folding. So this is a real-time trace of a ribosome translating, and as I said, 167 actually is seen here, and you see that nothing happened for a long while until eventually we begin to see some kind of transition, and that transition, you see it here clearly, corresponds to the attainment of the misfolded state. And then if you let it pass for another time, for more time, then you begin to see that the ribosome has continued to synthesize the protein, but then eventually you see a competition between the C-terminus here and the misfolded state. Here is the unfolded state, the C-terminus, and the misfolded state, and they are competing now. Okay, now what I want to point out here is how long did it pass between 167 and the appearance of the misfolded state. Okay, so we can actually measure the time between arrival at 167 and the observation of the misfolded state, and we find that actually that time is about, on average, about 63 seconds. So remember that in stall elongation complexes, which are equilibrated, the time to actually misfold was 1 millisecond, even on the surface of the ribosome. Now, but this is on stall equilibrated complexes, but now as the polypeptide is actually being born in real time, the ribosome somehow is preventing the attainment of the misfolded state for 63 seconds, which gives time for the rest of the protein, the C-terminus, to actually come out, and the C-terminus to outcompete the misfolded state by folding first, right? So essentially, what this is telling us is that co-translational folding is an out of equilibrium process, because the system is not, when the system is not equilibrated, it is essentially kinetically trapped. The polypeptide that is being born in real time is kinetically trapped by interactions with the surface of the ribosome or with the tunnel. And this morning I was talking with Silvia Cabañero, and she has data that actually is very similar to this, where she can see that actually interactions between the polypeptide and the tunnel itself actually are holding onto the onto the nascent chain, and essentially keeping it out of equilibrium by actually kinetically trapping the polypeptide. Interestingly, once it starts spontaneously, once it starts misfolding, it will just go back and do unfold, misfold, unfold, misfold, unfold, misfold all the time. Why? Because now it has equilibrated, right? And if we don't wait 63 seconds, but as soon as we get here to 167, we yank all these interactions between the polypeptide and the tunnel, the protein immediately starts misfolding, and then it will all the time misfold and unfold, misfold and unfold, misfold and unfold. So what this is telling us is that co-translational folding, in fact, depends on this kinetic trapping of the polypeptide being born and to prevent actually misfolded states in this case. So to conclude, not only the ribosome does not induce an end domain direct folding, but suppresses folding of the end domain, right? And stabilizes the misfolded state through both slower folding and faster unfolding, and actually keeps the nascent chain out of equilibrium by actually kinetically trapping it through interactions with the with the exit channel. That 1 minute delay that I showed you prevents misfolding and could allow time for chaperones to help the folding, or maybe further nascent chain synthesis by the ribosome for the whole protein to fold correctly. And then the other thing that I think we should actually point out is that part of some of this information that we are getting is because we are doing it in real time, and some equilibrium studies may actually miss some of the key dynamics that are important during actual co-translational folding. So I'm going to finish here. This is the end of the first story, and I'm going to start telling you about the second story, which is about the nascent chain, how the nascent chain can modulate ribosome activity. I just told you that the ribosome can affect folding. The question we are asking now is, can folding affect the activity of the ribosome? So the opposite direction, right? Okay, so as you probably know, there are many peptides that actually are known to actually possess rest sequences that interact with the tunnel in the ribosome and induce arrest at the peptidyl transferase center in the ribosome, okay? And these arrest sequences are known to modulate gene expression, protein production across a variety of processes. One of them is the Sec-M motif. The Sec-M motif is a 17 amino acid arrest sequence that binds to the tunnel and actually repress the transferase activity of the ribosome. And this Sec-M arrest peptide is important because it's involved in the regulation of the secretion machinery in E. coli, Sec-A in particular, which is the ATPase in the translocon. So this is the messenger RNA that actually codes for Sec-A, the ATPase in the translocase, essentially. It has this Child-Dalgarno sequence that is actually hidden in this hairpin, and then upstream it actually has this Sec-M section, and this 17th section here on the C-terminus, part of this Sec-M, which stands for the monitor, secretion monitor, actually includes and contains the 17 amino acid sequence that stalls the ribosome. So Sec-A is subjected to secretion defect response regulation, and the regulation actually is carried out by Sec-M, the secretion monitor, which encodes this 50 amino acid region upstream of the Sec-A gene, right? All right, so what happens is that when the ribosome is coming this way, it will actually find Sec-M, and it will actually stall the ribosome at proline 166, as you see here, okay, on Sec-M. Now, notice that because the ribosome has stalled here, the hairpin is gone. It has destabilized the hairpin, and it has exposed the Child-Dalgarno sequence ahead of the Sec-A gene, right? So now, if there is no sec-A in the cell, then the ribosome can bind in the chiral garno and will synthesize more sec-A. But if there is sec-A, okay, then this whole complex will actually move to the translocon, and the translocon will interact with the ribosome and the nascent chain and will de-repress the arrest. And the ribosome will quickly pass, the hairpin will form again, and there will be no synthesis of sec-A. So that's how the system is regulated. And so low sec-A activity, there is robust stalling, then there is upregulation of sec-A, right? But if you have low, okay, so that's what I said, right? So on the other hand, if you have high sec-A activity and the sec-A can actually repress the stalling of the ribosome, then the ribosome quickly passes, no stalling, downregulation of sec-A. Okay, so that's, again, to summarize. All right, so that's sec-M. Now, the first question that we asked in this study was, how is that the sec-A, which is a translocase, is an ATPase in the translocon, de-represses or liberates the arrest of the ribosome? One possibility is that just through allosteric interactions, it actually tells the ribosome, okay, let it go, and then the whole thing gets detached. Or it could be that it's mechanical because it's an ATPase, and therefore it may be functioning as a motor. So the question we ask is the ribosome stalling a sec-M release just by the generation of mechanical force by sec-A on the nascent chain, breaking critical tunnel interactions and then allowing for the ribosome to continue working. So what we wanted to test is this, see? And in the absence of the arrest peptide, normally what happens is that you have the tRNA aminoacyl in site A, then you have the peptidyl tRNA in the P site, and then you get the peptidyl transfer reaction, the formation of the peptide bond in A, then EFG binds, EFG induces translocation of the ribosome by one codon, and now the A site is available to add another tRNA. That's what happens in normal conditions. But when you have the sec-M system, right, you get stalled at proline 166, and now the ribosome cannot continue. And so the question that we ask is, if there is peptidyl transfer, what will happen, of course, is that then the bond is going to be formed, the peptidyl group is going to be transferred to the A site, then EFG will actually make it actually A accessible, right? When it is there repressed. This is what happens when it is there repressed. So we wanted to know if that process can actually be induced by force. In other words, if we grab the peptidyl and then we pull, can we actually make that happen, essentially just by applying force? Okay, we were trying to test this mechanical translocon hypothesis. In order to test that, we built this construct where you have the ribosome grabbed to a bit on a pipette, and then we have a ribosome that is stalled at the sec-M. We add this calmodulin N-terminus of calmodulin, and domain of calmodulin, and I will tell you why we do that, and then the DNA handle to attach that calmodulin to a bead, right, on the other trap. Okay, so in this case, the protein, the ribosome is stalled at the sec-M, and now the question is, can we actually make this peptidyl transfer happen just by applying tension on this polypeptide, right, on the nascent chain? If we can do that by just applying force, then it should be true that when we actually add EFG, and this has happened, and this peptidyl transfer reaction has happened, then when we add EFG, the ribosome is going to translocate and will have the A site available. And then we use this trick. We use puromycin. Puromycin is an amino acid that binds to the A site, And when it binds to the A site, it incorporates the puromycin at the end and breaks the tether. So if we see tether breaking, that is indication that the puromycin was able to bind to the A site, and that meant that actually the peptidyl transfer reaction had happened and that the EFG had allowed it to actually move to the P site, right? So we are going to see the breaking of the tether as a function of force, essentially in the presence of puromycin for this complex. Now, so this is the idea, right? We have a stall, we add puromycin, puromycin gets incorporated, the tether breaks. That indicates that the ribosome had actually carried out the peptidyl transfer reaction and moved by one codon. Now, the reason we use a calmodulin is because calmodulin has, at a force of about 7 piconewtons, it actually goes from folded to unfolded, folded to unfolded, right? So it has a unique signature. Look, here is folded, unfolded, folded, unfolded, folded, unfolded, right? So you can have this telegraph type of signature here, which is characteristic of calmodulin. So if we see that, we know that we have the right tether because the calmodulin is doing its thing, right? So that's to distinguish that we are seeing the right tether and not anything else. And then we can now raise the force, say to whatever, 20 piconewtons, for example, or 10 piconewtons, or 5 piconewtons, whatever, and then determine the breaking, how long it takes for the system to actually break. And what we found was that, in fact, as we increase the force from 10 piconewtons to 15 to 20, 25, 30 piconewtons, we could actually see a faster and faster breaking of the tether, indicating that, in fact, the force alone is sufficient to repress, to actually remove the stalling of the sec-M sequence. And so you see that we have the force dependence for the logarithm of the breaking of the tether. And notice that when we extrapolate to 0, we get to a value, and this dot here, this blue dot, is the rate of breaking of the sec-M spontaneously at 0 force in bulk studies. So you see that our data, our single molecule data, really coincides with the rate of spontaneous breaking at 0 force obtained from bulk experiments. Okay, so that's what we have, and so what we conclude is that mechanical force is sufficient to release the nascent chain-mediated arrest, which is what is called the mechanical translocon hypothesis. But now we want to ask another question, and is a question that immediately comes to mind. You have a polypeptide that is coming out on the surface of the ribosome, and the question is the following. Imagine that this polypeptide decides to actually fold. When it folds, in order to fold because of the surface of the ribosome, it has to actually generate what I call a steric exclusion force, because essentially the polypeptide has to do something like this, right? Has to, in the process of, since the polypeptide cannot go into the tunnel, what it will actually do is it will actually sort of, in folding, it will actually generate a force that will pull on the ribosome, and we wonder whether that force generated in the process of folding on the surface of the ribosome could de-repress the sec-M stalling of the ribosome. That's the question. If that is the case, then we could say that actually not only the ribosome controls in part the folding of the polypeptide, but actually the folding of the polypeptide can actually control the activity of the ribosome, right? And so in order to actually test this idea, we decided to use a protein that had never been in nature before. So we used TOP7, which is an artificial protein that was synthesized in the laboratory of David Baker. And so therefore, whatever effect we see is unlikely to be allosteric, but it's probably something that has to do with the folding of the protein. Then we have a linker, and we are going to generate a library of these linkers of different lengths between 4 amino acids and 28 amino acids. Then comes sec-M, which is we needed to stall the ribosome, right? And then finally a green fluorescent protein gene, right? And so what we are going to, here are a number of reasons why we choose top 7. It's not a nature existing protein. And then we are going to generate this library of various linkers to actually see what happens. So here is the idea. All right, so suppose that we use a short linker. When the linker is short, part of sec-M is obviously still buried inside the tunnel, right? So sec-M will not be top 7. Part of top 7 is what I wanted to say, not sec-M. Part of top 7 is still buried inside the ribosome, and therefore top 7 cannot fold, right? Okay, for the short linker. If the linker is very long, now it is completely away from the ribosome, and now top 7 can actually fold completely without generating any exclusion force. But if the linker is of intermediate size, right, then what will happen is that only a little part of top 7 is going to be inside the tunnel, and when it falls, it will generate a pulling force that actually will release the arrest, and then you will see the formation of green fluorescent protein. Is that right? Okay. So now we generated this library with different linkers, and then we transformed bacteria, and then we actually plated the bacteria and wanted to see if there were colonies that were fluorescent. And here you see that many of the colonies were not fluorescent, but some of the colonies were fluorescent. So then we actually sequenced the colonies that were fluorescent and wanted to see what is the size of the linkers. And when we did that, you see that the size of the linkers that actually led to fluorescence were between 15 and 21, 22 amino acids, which span more or less a good part of the tunnel in the ribosome. And when we sequence all the linker sizes for all the colonies, regardless of whether they were fluorescent or not, we regain our original distribution of linker sizes. So this essentially is telling us that for a particular size of linker, in fact, the folding of top 7 actually probably led to the actual release of sec-M, and the fourth generation was enough so that actually the green fluorescent protein could be synthesized. And so folding exerts a force capable of releasing native change-induced arrest. And now you may ask, how much force does top 7 folding exert on the nascent chain? In order to answer that, what we need to do is grab top 7 and fold it and refold it and see at what forces it unfolds and it refolds. And so now, by the way, I just wrote here 2 things. One is that when top 7 falls near the exit tunnel, it will fold against an exclusion force that it generates when it tries to fold. But that force, because of Newton's third law, is also the force against which the protein is folding, right? Because it's action-reaction, right? And also the protein, top 7, has to be not only able to fold against this force, but remain folded for enough time to be able to release the arrest by sec-M. Okay, so first we pull sec-M, and you can see that the protein unfolds and refolds, unfolds in red at different forces stochastically, and then refolds in blue, right? Again, at different forces, again, of a stochastic nature, right? Okay, so then out of this you get a distribution of unfolding forces and a distribution of refolding forces. And here you see the distribution of folding and distribution of unfolding. Olga Dudko at UC San Diego actually showed that out of these histograms it's possible to obtain the lifetime of the folded state and the lifetime of the unfolded state through this expression. And from that, we can get, once we have the lifetime, the inverse of the lifetime gives us the rates, right? And so it's possible to actually obtain the rate of unfolding of top 7 and the rate of unfolding of top 7 and the rate of refolding of top 7 as a function of force. Now, you notice that these curves cross at this point. At this point, you see that the rate of unfolding is equal to the rate of refolding. So at this force, which is about 12 piconewtons, the equilibrium constant of this protein is exactly 1 for unfolding and refolding, right? And then, not only that, at this force, we find that the protein remains unfolded the same amount of time unfolded that it remains folded, and that is 28 seconds. So at 12 piconewtons, the protein, top 7, can refold and stay folded for 28 seconds, pulling on the sec-m before it unfolds. And then it unfolds for another 28 seconds, and then after 28 seconds, it actually again starts pulling for another 28 seconds, right? Half a minute. Okay, so we want to know what is the force at which top 7 did this work. And so here is the kinetic diagram. The protein can go from unfolded to folded on the surface of the ribosome, and that is force dependent, of course, because it's creating this exclusion force against which it is actually folding. Now, the sec-m before the protein unfolds can spontaneously release at a rate k0, which I told you in the blue dot that I showed you before. We know that value, and we also know it by the extrapolation. This data I have just shown you, the rates of unfolding and refolding as a function of force, and there is this rate of release, which I actually showed you before. The rate of release as a function of force is this. Here is the spontaneous rate, k0, and these rates we have just determined as a function of force for unfolding and for refolding, right? So we essentially we know all the rate constants now for this system, and we can calculate what is the effective rate of relief, which can be either the sum of the spontaneous rate plus the force-dependent rate, and we obtain this expression. And when you plot this expression in terms of all the values that we know, that we have determined, we get this funny-looking curve here that makes a maximum. Why does it make a maximum? Because, of course, at low force, at 0 force, the protein actually is, the release of the sec-m is occurring at the spontaneous value k0, right? Then, as the force that against which the protein is folding increases, it also increases, and it makes a maximum here. And then at higher forces, the protein again comes back to this value, because now at this force, the protein is most of the time unfolded, right? Here is not generating any force. Here is actually making the maximum because it's folded and it's exerting force. But beyond that force, the protein quickly becomes easy to unfold, and now the protein is unfolded and it cannot exert any force to release the ribosome. And so you can calculate what is the probability that the ribosome unfolds by this path and not this path, which is given by this expression. And you can see that the maximum occurs at about 10 piconewtons. So top 7 actually can exert up to 10 piconewtons of force and release sec-m. At 12 piconewtons, it can stay folded for 28 seconds. At 10 piconewtons, it probably can stay folded maybe for 40 seconds, and so it's holding onto it and finally is able to actually release the arrest. So to summarize, a protein folding at the ribosome tunnel can exert force capable of releasing nature change mediated translational signaling. Force generated by folding could help tune ribosome activity to transitions, folding transitions in the nascent chain, and other co-translational process could generate force to modulate translational rates, because maybe a chaperone will bind to a ribosome to a nascent chain when it is coming out, and the binding of the chaperone may actually generate a force that could actually affect the kinetics of translation. So I want to just acknowledge the people that have actually done the co-translational studies were done by Lisa Alexander, a terrific graduate student, Daniel Goldman, also a graduate student, a great graduate student, and Liang Mingwei. We, postdoctoral in the lab, and the SEC-MRS was done mostly by Christian Kaiser, Dan Goldman, and was one of the last pieces of work that I did with my ex-research advisor, Nacho Tinoco, before he passed away. So let me thank you for your attention.
Okay, so we have a few minutes for questions.
Hi.
I don't know if the mic's working. Have to speak up.
I speak really loud, so I
Yes.
This was great. I just have two questions regarding the first part. That delay in folding, does that mean that the protein is in a nuclei in which the folding can start, or it's just, it's like there's going to be a core that is going to lead all the folding of all the regions that weren't folding before. That's why this delay is happening. That's the first part. And the second part, when you're talking in this second talk about the force you have to do, what is the relationship with the RNA structure that you have there? Because I imagine that when you are folding this way, then you have to move RNA, so that means that the RNA that is outside, the ribosome needs also to unfold to be able to come in. So is this related to the RNA structure too?
Yeah, let me answer first the second question, okay? In that experiment, we grab the ribosome and then we just grab the polypeptide chain. Mm-hmm. So we never exert any force on the messenger RNA. The messenger RNA is not touched at all. So it is only that we are actually pulling on the polypeptide chain, and we are asking whether force is sufficient to actually release the arrest, mechanically, the arrest. But we don't touch the messenger RNA at all,
Okay,
at all.
So if my question is, I imagine, because RNA structure can stall the ribosome in
It can.
So if I have a more complicated structure that can by itself stall the RNA, does this process can happen the same way? Like, do you think the mechanism?
No, these are a different process. So RNA can actually form a hairpin, and certain hairpins, the ribosome actually can open hairpins. It has an intrinsic helicase activity. And we have studied the helicase activity by single molecule, by this type of experiments already in the past. And if you actually increase the number of GCs in the hairpin, you can actually make the ribosome move very slowly. If you start increasing the percentage of ATs, the ribosome starts moving faster. That is by a completely different mechanism. It's really a mechanical barrier that the proteins at the entry site of the messenger RNA in the small subunit, which are S3, S4, and S5, have to actually negotiate to actually open that juncture, the hairpin, and that is completely a different thing than arrest induced by the polypeptide that is being, the nascent chain that is interacting with the tunnel as it's being synthesized. Your first question was about the first part, which is how is that the protein actually, how is that the ribosome actually prevents for a whole minute the protein from misfolding, right? We don't know the answer to the question in the type of detail that we would like to know. For example, is making these contacts, but maybe with the wonders of cryo-electron microscopy, we may be able to actually freeze quickly complexes before they equilibrate, and we may be able to actually see which contacts actually are, and then let it equilibrate. Let's wait 5 minutes, equilibrate, and freeze them, and then compare and see, okay, look, this is how it is before it equilibrates, this is how it is after they equilibrate spontaneously after 1 minute. So I don't know if it is possible these days to actually make that kind of of temporal resolution with cryo-EM, but I think probably it should be possible to design an experiment to distinguish these two things, and we may be able to actually see a different type of structure when it is before equilibration and after equilibration. So, but I don't know the answer yet, really.
Thank you so much.
Sure. Thank you.
Great talk, Carlos. I was just wondering on the first story,
Yes.
if you looked at the effect of molecular chaperones on this huge delay that you are seeing. Are they changing the situation or not?
Yes, no, that's a great question. I think that actually it would be great. We haven't actually used any chaperones at this point, but I think it would be really great if we could actually then do the same type of study, but now in the presence of the chaperone, and see if in fact the kinetics, how the kinetics changed. And it's possible that my intuition is that if anything, the chaperone is going to actually favor the folding of the C domain. And once the C domain falls, the misfolded state cannot happen, because then the only thing that is left to fold is the N-terminus domain, which loves to fold once the C-terminus domain has folded. But it would be great to actually use a chaperone, so maybe we should talk about that.
Okay, thank you.
Thank you, Sylvia.
I don't know.
Would you mind going back to slide number 12?
It's slide number 12. Oh, great. Thank you.
Most of the numbers, I really this slide is talking about we were talking about the on pathway versus off pathway intermediates.
Oh, yes. Okay. So, sorry. Here. Yes?
Yes. So the evidence that you're on pathway is that you go from unfolded, you hit the intermediate, and then you go to fold.
Yes. Yes, that's right.
And I guess when you do this, 100 or on a bunch of different proteins, or 100 different iterations on the same protein, do you always see the intermediate before you go to fold it, or do you see some statistical distribution?
I tell you, in the cases that we have studied, where we have done this type of analysis, for example, RNase H, a protein that actually falls through an intermediate, calerithrin falls through this intermediate, we either see the intermediate and lasting for a while, or we sometimes don't see the intermediate and we go directly from unfolded to folded. But if you now plot the lifetime of the intermediate, which is actually given by, if you plot the lifetime of the intermediate, which is given now by this little piece of time, right? Now you see that actually there is a Gaussian distribution where there is some that are so short that the optical tweezers cannot detect, and some are very long, and some have some value in between. And so, but when we actually have seen misfolded states that are off pathway, then you never see from the, let's say now this is the misfolded state, this intermediate, we never see it going to the folded state, but what we'll see is actually going first to unfolded and then to the folded state. So then this is an off pathway intermediate.
So you can imagine some intermediate where once you're in the intermediate, the potential energy landscape is that you're just as likely to go back to the unfolded as to the folded. Have you ever seen calculus system like that? Where you could go to the intermediate, but you don't have to go to the intermediate?
Oh, it doesn't have, this is one case, but sometimes we see going, well, here you see, it's unfolded, folded, and it went to the unfolded again instead of going to the folded. And then after a while, it went to the folded. So this is exactly that case, right? Yeah. It would have been more dramatic if I show you an example where this time lasted very little. Then you could see that actually it either goes one way or the other. But that's the property of the on pathway intermediate, which actually always has two possibilities, going back to the unfolded state or go back directly to the folded state, right? Yes.
You said this protein binds calcium.
Yes.
Does calcium affect the kinetics?
It does, and all of the work has actually been done in the presence of calcium. It speeds up folding in the presence of calcium, yes. And it can actually, sorry, I was told that I shouldn't move too far from this line. Okay. Yeah, so calcium affects it in a more or less monotonic fashion, probably because it organizes the loops. These EF hands are helix loop helix, and the loops bind calcium, coordinate calcium. So it probably helps to actually form the EF hands, organize the EF hands. Yes. Yes.
Are the intermediates primarily rearrangements of tertiary structure? So in other words, you fold the alpha helices and they stay folded down, right? And then you're rearranging how those fit to each other? Or are the intermediates actually unfolding the secondary structure as well?
Okay, so let me say, in this particular case, we know, as I showed you in the talk, that the intermediate corresponds to the folding of the C-terminus domain. And the C-terminus domain has two EF hands, and that's helix, loop, helix, helix, loop, helix. So it's four helices and two loops that actually are folding together, and that constitutes the intermediate in this particular case. Now, your question is more generic, more general. What you are saying is your method would allow you to tell what part of the protein is being unfolded if it is not the whole.
Or I fold an EF hand and I fold another one.
Yes.
And how I fit them together.
That's right, exactly. Okay, so let me tell you what the situation is with that. We are actually doing. Okay, so as you know, there is two basic models right now that are competing to try to explain how proteins fold. The funnel model, which everybody knows, which actually is in all the books, that says that the protein can actually start anywhere in its disordered unfolded state and start coming down the free energy surface because all roads lead to Rome, right? I mean, so since it is a funnel, you can actually come down by a myriad of possible processes all the way down because they will actually be, as soon as you start moving down from anywhere in the funnel, you are actually getting closer in free energy to the native state. I don't believe that's true. And I don't believe that's true because what that means is that the information encoded in the sequence of amino acids is equal throughout the amino acid, and that nucleation of the folding can start anywhere. So you, for example, at one point, this part of the protein may organize a helix beta sheet structure, here a helix loop helix, here, and that any one of these pathways is possible because it's part of is forming already the final folded state, and it's decreasing the free energy of the system. I don't think that's the way it happens. I think it's impossible that that's the way proteins evolved. The alternative model, which is the one I subscribe to, and that we are trying to test and demonstrate, is the fold-on model. The fold-on says that the information encoded in the sequence is really present in runs of amino acids that are 20 to 25 amino acids long, and that Walter Englander has called foldons. I was saying it in Spanish, foldons, okay? And these foldons contain the information, and they are sequences that are very prone to actually attain secondary structure, and that the process of folding of a protein actually follows a well-defined path in which one foldon initiates the folding, maybe this other folding takes advantage that this has formed and actually folds on top of it, and so on and so forth. And that makes a lot more sense, in my opinion, than the other information. And then what that means is that that what nature had actually selected is the use of these units that are folding units, that are discrete folding units that actually are combined in different ways, in different sequences, but that the information is not in the amino acid, at the amino acid level. The information for folding is in these units that are longer than one amino acid, 20, 25 amino acids. If I tell you, how do you design a house? You don't tell me, well, you start actually with this brick and this other brick and this other building brick, because that's not how you think of a house. How do you design a house? You say, well, it has to have a living room, it has to have a dining room, it has to have a bedroom, it has to have a bathroom, and those are the units that make a house. Now, it's true that those units are made out of bricks, amino acids. But really what determines what a house is really these larger units that actually are part of the design of the protein. And so once you actually have folded one domain, the C domain, for example, I would call that the first floor of the house, okay? Now you need to get the folding of the second floor, which is the N-terminus domain, for example, in the case of caleritrine. So I believe that it is possible to test these ideas, and we have actually now, we are doing experiments right now where we are using the optical tweezers to study a particular protein and determining in which order the different parts of the protein are actually folding. And I'm happy to tell you that actually the protein that we have just looked at and we have completed folds by a single pathway always. Always the same part of the protein falls first, then the next part, then the next part, the next part. Now you may say, how come you were able to do that? Because here we see full transitions, not individual part transitions. Well, we played a trick. We said, in the different parts of the protein, we're going to put pieces of glycine, long runs of glycine, so that when that part unfolds and that part refolds, we will see that actually that's the part because it has been lengthened by the glycine length that we have added. And by doing that, we've been able to establish which part of the protein is folding, and the protein always folds by one path, not by an infinite number of paths as the funnel says. Now, it is a downhill, so you could actually think that it's a cross-section or the funnel. If you imagine the funnel, just take a little piece of the funnel. That's true, but not the whole funnel. That cannot be. Yes?
It's fair to say that the fold-on model relies on the ribosome, and that if you're just looking at refolding in vitro, it may be the funnel model. Is that fair?
Well, we are doing this in vitro, not in, our studies are in vitro, but rather what would be interesting is to ask the question, how is that the fold-on model is modified by the ribosome? That is an absolutely fair question, and it would be foolish of me to actually try to speculate about the question that I have not thought too much about in front of so many people. So, but I think that it would be very interesting to think about how the ribosome may affect the folding. But for example, in the case of caleritrine, if I may say so, the N-terminus doesn't fold until the C-terminus come out. So the ribosome is not really affecting it's almost like the ribosome is saying, nobody fold here until I say we can fold, right? I mean, and so then maybe when the C-terminus fold, the N-terminus is following its own path of folding, right? Yeah.
Great. Well, thank you so much for
Thank
being
you.
here. Let's give Dr. Bustamante another round of applause.